All right, sweet. All right, we're recording, we're rocking and rolling. It is time to go over activity one. We're going to talk about some inflammation, some antigens, and more things. Hopefully this will start to help you all put the different pieces together. Please make sure to stop me and ask questions as we're going on, since if you have a question, I guarantee that probably like everyone else has a question, because this is just really confusing and there's lots going on here. So please interrupt me and ask questions if you have them. All right, number one, how do interferons and the complement system work? So these are separate things, but they're two important signaling things. Who would like to explain number one?
I can.
Okay, thanks, Jenna.
So interferons, I mean, the name helps because they interfere with viral infections by inhibiting viral replication. So pretty much they stop them where they are. And then the complement system is activation of a series of proteins that result in a cascade of biochemical reactions that help stop the infection.
Awesome, yeah. So we're having two things right here that are stopping the spread of an infection, but they work a little bit differently. Which one of these is part of stopping a viral spread?
Interferons.
Interferons. And are the interferons a type of cell, or are they a signal that go between cells?
The signal.
Right, they're the signal. And Cole, could you explain a bit of how that signal gets passed around and what it does?
Yes. So the signal is kind of like when I think it said when a macrophage is infected.
It's just like when any cell is infected.
Oh, when any cell is infected, it will just send out like chemicals alerting other cells that it is infected, and the other cells will then, I think, release their own chemicals that are meant to prevent the pathogen from getting in or from infecting it.
Yeah. And what happens if the cell, a separate cell, is already infected and an interferon signals to it? Then what will happen?
To the infected cell?
Mm-hmm.
Will they die? Lysis?
Yeah, exactly. They do apoptosis, which is a throwback from last semester, but it's a fancy term for self-destruction of a cell. Yeah, really well said, Cole. So if you have one infected cell, it releases interferons, these little proteins. It's kind of like a ship going down, and they shoot off fireworks as a distress signal. They're like, hey, something is sinking our ship. Get ready, it might sink your ship too. And so then other cells that are not infected stop producing proteins, they stop producing RNA. So that way, if a virus were to infect them, all of those protein and RNA producing things are shut down. So the virus would not be able to hijack them and make more viruses. And then in other cells, an interferon might signal to that are already infected. The cell recognizes, oh, I already am infected. I'm going to commit apoptosis, self-destruct, and then then the virus won't be able to spread. Does that make sense? Mm-hmm. So the interferons are signaling molecules, not cells themselves. And then the complement system, as Jenna said, is really cool. It's a way of stopping the pathogens by destroying it themselves. Do any of y'all know what the actual mechanism, so you've got the pathogen and all the complement proteins gather onto it. What do they do to it to actually destroy it?
I don't know.
Don't they do like phagocytosis or whatever, like engulf it and then put in a lysosome, destroy and then expel it? Or is that a different thing?
I think that's a different one, although it can't be flagged for it. But
are you talking about how they destroy the RNA?
That would be more something with interferons.
Just kidding.
But it's a good guess. Do any of y'all remember hearing about the membrane attack complex?
Yes. Doesn't it like put a pore into the complex so everything rushes in and then or rushes out? I can't remember.
Yeah, exactly. You're spot on. So outside of the pathogen are those little proteins that our cells recognize, and then these little complement proteins recognize them and bind to it, and they attract more and more and more different kinds of complement proteins until they make this whole thing called the membrane attack complex, which it's not so much that it's attacking the membrane so much as it's making a little hole in it. And that hole or that pore then allows all sorts of water and salts to rush into the cell, and then it swells up and explodes so that the pathogen dies. So that's like the end goal of complement is destruction of individual pathogens by making those pores in them, and then water and ions rush in and they explode. Nice. Way to go, Regina. Sweet. Okay, number 2. How does inflammation occur? Who had numero dos?
I did.
Oh, okay. Sweet.
So inflammation is for like minor injuries, like the first level of response. And first, macrophages discharge cytokines. I'm sorry, I can't pronounce half of these things.
Yeah.
And signal molecules. Those molecules are recruiting neutrophils, and then mast cells, these other cells, release histamines, which are histamines. And what these do is they like dilate the capillaries, so then the like recruited neutrophils can like come out from the like capillaries, so they may basically make it permeable, and then these neutrophils are able to digest the pathogens that come in like in the cut.
Yeah, awesome. That is all of it wrapped up into one. Yeah, the main goal of this is that you're getting a bunch of things to come to one spot. So especially when you have those mast cells secreting the histamine, then the blood vessels dilate. And then aside from cells, what other sort of defensive things do y'all think our blood can bring to a certain location?
Blood clotting?
Oh.
Yeah, like platelets.
Platelets, yeah, if there's a pore. What about what proteins might they bring to help fight? Hint, we just talked about it last question.
Peptides?
Yeah, peptides, antimicrobial peptides, and also complement proteins. Since all of those complement proteins are flowing around in the blood, and you want those proteins to get to the infection, all you have to do is direct blood flow there, and then they'll do their thing, they'll fight the pathogen, and you'll be okay. What do y'all think could be one problem, though, in causing inflammation? Or not even one problem, but a handful of problems. Or is there not an issue? Do we want infinite inflammation?
Isn't it like systemic or systematic like thing where then it just like swells up too much and like basically infects your entire body from that site and then like just spreads?
Yeah, that could be one issue. If you over inflate, or if you're overly inflamed, you could have a swelling that could lead to all sorts of damage and could possibly end up with more of a spread of a virus. Because you have all sorts of blood flow there, it's possible that the cell then hops in that and then gets somewhere else. There's also all sorts of diseases that are caused just by inflammation. And if you think about like allergies, like that's why many of us take antihistamines like Claritin. It's because you want to stop the histamines from doing their thing and causing massive inflammation in your nose or wherever your mouth, if you're breathing from that too. So anyway, okay, moving on to 3. What are the 2 main cells of adaptive immunity, and generally, how do they work? Who had 3?
I did.
Sweet.
So the 2 main cells would be the T cells and the B cells, and both of those are lymphocytes, so they'll go to fight infections. And each one is a little more specific, with the T cells being the mature lymphocytes, and then the B cells being the ones in mature bone marrow.
Yes, exactly. Where they mature is what gives them their name. So since T cells grow in your thymus, which is like somewhere around your throat, and I think in the PowerPoint there's a photo of no, sorry, I'm thinking of thyroid. Your thymus is, I think, near your heart, actually, because there's a picture of a mouse which has its thymus sitting on top, and it's huge. It's actually really large in certain animals. But anyway, yes. So T cells grow up or mature in the thymus. B cells mature in the bone marrow. And then, how specific is the binding with B and T cells? Are they kind of specific, super specific, not really that specific.
It's probably
Very
specific, or it's specific, right?
Is it super specific or
the
just kind of specific?
Probably super.
Yes, they're like super, super specific. And that's why they're a part of your adaptive immune system. So remember we talked about the two main branches. There's the adaptive immune system and the, what do you call the other one?
Innate.
Innate, yeah. So your innate is like very broad in general. That's like the complement proteins. It kind of attacks what a lot of pathogens have. It's not super specific, but it's effective. The adaptive immunity has very adapted responses that target and kill very specific pathogens. So since the B and T cells are part of the adaptive immune system, you can think of them being as super specific in what they bind to and in what they attack. But we'll get into that soon with the receptors. Number 4, what are the 4 main characteristics of the adaptive immune system? Oops.
Okay, so the 4 main characteristics. So the first one is immense diversity of lymphocytes and receptors. Obviously, lymphocytes are both B cells and T cells, and like there's, as far as the diversity, there's a lot of different kinds of receptors. Like we just said, they're very specific for different types of pathogens. So that gives them their huge diversity. And then self-tolerance, which is a lack of reactivity against an animals and molecules and cells, which in more simpler terms, like the B cells and T cells aren't going to act against your own body. Like they're developed so that they can attack foreign particles and not your body's own, not any objects that belong to your body. And then B and T cells proliferate after activation. So after they activate B and T cells, it's a whole process where it's like they divide or they clone themselves first, and then those clones become either memory cells, which will be used for second response if that pathogen ever comes back. And then they'll also become plasma cells, which will secrete antibodies to attack the pathogen. And then finally, the last one is immunological memory, which is like how I said earlier. It's basically when if you get sick and you're able to fight it off, and let's say you get that same pathogen again, your body's going to react faster to it.
Nice. Yeah, super well said, Will. Yeah, I think, and these also, they show up in one part of the PowerPoint. It's just 4 listed things. So I would just make sure to memorize these as the 4 things. We were talking about in some of the groups. In reality, there's a lot of different ways you could look at this, but this is what Dr. Fernandez has labeled as the 4 things. So know it, know their parts, and how things fit into it. Jenna, did you have a question?
No.
Oh, okay. Sorry, I thought I saw your hand. Any questions on 4? Okay, sweet. Thanks, Will. Number 5. Oh, we're getting into the fun stuff. Describe the structure of antigen receptors. Who had 5?
I did. So we didn't really get a lot, but like the definition of what an antigen receptor is that there are proteins that allow T or B cells to attach to the antigen on helper T cells, and then just that they bind to a specific part on each antigen.
Nice, exactly. And what do you call the specific part of the antigen that you're binding to?
Isn't it just antigen fragments, or is that different?
That's true. You could like fragment an antigen, but if the antigen is this whole protein or something like that, what is the one particular spot where the antigen receptor is binding to it? This is open to everyone.
Isn't it the epitope?
Yeah, the epitope. It's just like the one tiny part of the antigen which is coming from your pathogen. That epitope is what's actually like the exact spot where you bind to, which could be just 1 or 2 amino acids, or it could be a handful of them depending on the shape and size of your receptor. And then, okay, another thing is that these, and y'all don't have to know all the background on this, The receptors are made of light chains and heavy chains. That has to do with a very complex genetic process of how these things are made. You don't need to know what light and heavy mean here, but just know that those are also parts of it. And then there's also some other specific regions of it. Do y'all think that we want to make receptors that are all completely constant, all the exact same?
Nope.
No, right? We want some variability, right? So as a part of that, you're going to have some sections that are variable and some sections of constant, that of the receptor that are constant. Do you think the part that binds to the antigen is the variable part or the constant part?
Find the constant. No.
Variable part.
Some constant, some variable. If your constant region is the same on every single antibody, and that's the part that you use to bind to different antigens, how well will it bind to different antigens? Not quite as well, right? It'd be like if I used my key as the thing that binds to something, and I tried to use that on my door and my neighbor's door and the house across the street and my car door. It might work well for one of them or kind of for all of them, but not really well enough. So instead, you have your body has the very tip of those receptors. It's very variable. And the important part comes when you're making the receptors. But imagine if you were trying to, for good reason, break into someone's house and you wanted to have a key that fit the lock just perfectly. You could just have one key and make 1,000 of that exact key and try each of those same keys in the lock. Will that work? No. Probably not, unless you got really lucky. What you'd more likely do is you make 1,000 different keys and you hope that one of them happens to fit the lock just well enough. And then probably after you find that key, you're going to make a bunch of keys that are really similar to that one, but even slightly different from that. And maybe one of those will fit even better in the lock. And if you keep doing that, you'll eventually get a key that actually fits perfectly. And then you can break into the house and hopefully do whatever good thing you're intending to do, or whatever. But that's kind of what happens when you make receptors and you're trying to bind them to an antigen. You keep making slight variations on the variable section to hopefully make a better fit so it can finally bind perfectly. Does that make sense? And that goes into the memory too, because as you develop better keys, then when the pathogen comes back in 7 years or 10 years or 15 or 20 years, you already have a key that fits it or fights it perfectly, and you don't have to go through the process of developing that key, which takes 7 to 21 to even longer, a couple of days or weeks, depending on the virus or whatever. So that kind of gets in the memory. Okay, sorry, that was a lot of me explaining things. What questions do y'all have on 5?
So you don't think that we need to know anything about the light and heavy chains other than the fact that they're a thing?
Right. If y'all want, I was talking to someone about this. If y'all want, I can send you my old lecture slides from when I took immunology, and you can learn all the deep genetics behind it. But I don't know that Dr. Fernandez cares or would bother testing y'all on it. So, okay. Hopping on to 6. Are antigen receptors and antibodies just the same thing? What's going on here? Who had number 6?
I had number 6, and I said that they're kind of similar, but antibodies they lack the membrane anchor. That's the main difference between them. And then the definition for the antigen receptor is that they're proteins which facilitate the binding of a B or T cell to an antigen, and then this produces recognition. And then antibodies are proteins which are the soluble forms of the receptor, and they have the same Y-shaped structure as B cell antigen receptors, and they're free floating, and I think they're produced by plasma cells.
Boom. Yeah, that was super well explained. Basically, you start off with your typical B cell, and it's got your little Y-shaped thing on it. That's an antigen receptor. And at some point, that B cell gets turned into a plasma cell. there's not really much of a change that goes on other than it starts secreting those receptors, which we now call antibodies or immunoglobulins, which get abbreviated as IG. So sometimes you'll see little like Y-shaped things floating around with an IG next to it. That IG stands for immunoglobulin, which just means it's an antibody, which is basically the same thing as a receptor, as an antigen receptor, but it's off the cell now. So lots of names for the same thing, which I think sometime in activity 2, we'll go through the 5 different types of it, but not in too much detail. So hopefully it won't be too crazy. Cool. Thanks, Tila. Number 7, how do T cells work? Oh boy, how is this different from B cells? Who had 7?
Except I don't have like very in-depth response. So, but T cells bind to antigens and then they go back to the lymph node to produce a further body response. And they can activate B cells and other T cells. Like that's an example of what helper T cells do. They basically like recruit everybody to help with it. And then B cells are activated and release antibodies. And again, they bind to the specific epitope.
Nice. Yeah, so T cells do some kind of funny things, but they also have receptors. They also go to lymph nodes. Yeah, there's a lot going on here. Real quick, a couple like smaller details before we go to the MHC stuff. T cells have alpha and beta chains, whereas B cells have heavy and light chains. So that's just like one difference to note between the two. T cells have alpha and beta chains. B cells have the heavy and light chains. But the T cells, as you can probably figure out, still has the variable and constant regions because you want to have that variable region to bind really well to the antigen. Real quick, since a lot of people are asking about the whole MHC thing, I'm going to give y'all a mini spiel on that and then keep going if that's okay. Because it's kind of confusing with all the macrophage stuff. So, and with T cells too. So if you imagine that I am a macrophage, which means I am one of your cells on your side fighting off infections. The MHC, which stands for Major Histocompatibility Complex, that's a long name, just call it MHC, please, is like an extension. It's like an arm on the outside of the macrophage. It's a complex that is part of the macrophage sticking on the outside. So my arm is the MHC, my body is the macrophage. What that does is let's say you have this antigen here, the MHC holds on to the antigen and presents it to T cells. So I, the macrophage, have the MHC, which is part of me. The MHC holds this antigen, so that way if, I don't know, let's say Viraj is the T cell, I can go up to Viraj, the T cell, and go, hey dude, this is the antigen. This is from the pathogen that I want you to destroy. And then Viraj, being a T cell, particularly we'll say a helper T cell, is going to bind to the antigen and in a second binding, bind to the MHC. So that's why T cells bind to antigens and the MHC. Then Viraj is going to go, okay, neat, and he's going to take this antigen, imagine just going through the computer, he's going to take it to the lymph node, and then your body's going to start to produce more cells that will respond to that antigen. So the MHC is a part of your body that holds, or part of your cells that hold on to the antigen.
Are all T cells helper T cells?
Perfect question. Are all T cells helper T cells?
No.
No. There are like 1,000,000,001 types of T cells. Not actually, there's probably more like 15 or 20. But generally speaking, there's a couple more important ones. The helper T cells are really great because they go and they help turn B cells into plasma cells.
Okay.
So they help get B cells to start secreting the immunoglobulins or the antibodies, which is really important. Helper T cells also go help other T cells called cytotoxic T cells. So you have helper T cells and then cytotoxic T cells, which according to their name, they're literally toxic to cells. Those cytotoxic T cells go up to your cells that have been infected with a virus and cause that all to get destroyed. And that way the virus can't use that cell and keep multiplying. But for the cytotoxic T cell to destroy your infected cell, it needs help from the helper T cell.
Okay.
If
So the
it didn't,
helper T cells basically just help other lymphocytes or they help bring other lymphocytes to infected cells, basically.
Yeah, that's a great way to put it. They help get cytotoxic T cells to infected T cells, and they help activate B cells and plasma cells.
Okay.
And they're really important because if they didn't help with those cytotoxic T cells could go around and kill all of your cells, and that would not be good. So, okay. Does that help explain kind of the overall picture of T cells and their interaction with B cells?
Yes.
What further questions do y'all have on them?
So the helper T cell just acts as like a director, and it just kind of tells everyone what to do.
Pretty much, yeah.
Okay.
And these are a little bit generalizing, a little bit broad, but explain, I think, at this level well. If you go on to a higher level, you'll find out the things I'm saying are not quite as perfectly specific as possible, but we don't have about 4 hours of lecture time to go through all the details. So these are like some, I think, good and helpful generalizations that I'll explain things mostly well. What other questions do y'all have? Word of T cells? Okay, ditto. We'll move on to 8 then. Oh, just kidding. There's more T cells. Never mind. 8. How do we make sure that T cells can attack pathogens but won't attack our own cells? So basically, how do we make T cells that are going to not attack our own cells but will attack other ones? Who had number 8?
We had number 8. So T cells, they go through a selection. So the first selection is a positive selection where the helper T cell will bind to the antigen presented by the MHC of a cell, and just to make sure the T cell can bind to the antibody. And then there's a negative selection that will, I think, kill off T cells that bind too well to other cells, so to prevent that T cell from killing our cells.
Nice. Okay. Yes. I'm going to add a few details, but that is absolutely the general process.
Okay.
Think about it. If you were making, let's say you're making an army to fight off the pathogens, and you're locking and loading, and you're giving your soldiers a bunch of weapons, and then you just say, okay, go and destroy things, but you don't tell them who to shoot at. They're probably going to shoot some of your own people, and that's really not good. You also want to give them a gun and then make sure that that gun actually works. Otherwise, they're not going to be able to do anything either, right?
Yes.
You want to find some sort of balance between equipping them to do stuff and making sure they don't attack and destroy everything. So the first step when we're making these T cells is we go through positive selection, like Cole was saying, and you make sure that your T cells can bind to the MHC on macrophages. So this is like, all right, we're making our T cells. We need to make sure these guys can actually interact with our macrophages and actually work together with them to then stimulate an immune response. Because if they don't work with the MHCs, they're just not going to do anything. They'll never bind to the MHC and bind to the antigen, and they'll just be useless. So positive selection is like, all right, can you work at all? Does this work? Does your weapon function, basically? Then after cells make it through positive selection, then they go through negative selection. So, okay, we know that your gun or your weapon is whatever is working. Now let's make sure you're not so crazy that you end up shooting your own people or attacking your own cells. The way that works is basically you go through and you check that T cell receptor and you make sure that it is specific to the MHC and it's specific to pathogenic proteins. Because otherwise, if the binding is strong but really general, it could end up binding to your own cells and telling your immune system to start destroying your own cells. And there are actually a number of diseases, autoimmune diseases, where that's what's happening. You don't have any sort of self-tolerance. Your immune system cannot tolerate itself, or like self as in your own body, so it starts attacking yourself. And that's not good. So first you have positive selection to make sure that your T cells can bind to the MHC and do things. Then you have negative selection to get rid of those T cells that bind too strongly. Again, the details of this are really complicated, and it all happens in your lymph nodes in lots of cool and complicated ways. But for now, just I would just take it that that's how it works. If you want to, I'd love to stay on afterwards and talk to you about it, but it is probably too long to explain at this moment. But do y'all have any questions in general about the process of like negative and positive selection? No? Does that make sense how you have to have like one step to make sure it works and another one to make sure it's not working too strongly? Okay, that's kind of like the general thing here. All right, we're gonna jump on to 9, and then we'll probably save 10 for activity 2 if that's okay. And then we'll go from there. Who had number 9? Anyone? Did anyone get to 9?
I kind of got to it.
Okay, what do you have?
Okay, so you have the spleen, which filters blood, and then the lymph nodes are like an immunologic filter, and then dendritic cells reside outside of the lymph node to stimulate adaptive immunity. That's all I got to so far.
Nice. Yeah, those are like some of the big parts of it. So as you're having your blood go throughout the body, it goes to your spleen. And just as Lauren was saying, that kind of filters out some things of it. And then when you have your lymph getting pumped through your body, it goes to the lymph node. And the lymph node is going to be a filter that cleans it or checks it for pathogens. Dendritic cells are one we haven't talked about as much. You can think of them as pretty similar to macrophages in that they engulf pathogens and then they present the antigens later. What it means when, I think this is in the slides or in the voiceover, that it stimulates adaptive immunity. That's a fancy way of saying that the dendritic cells, after engulfing something, after picking out an antigen, take the antigen to the lymph node and say, hey, B cells, T cells, here's an antigen from the pathogens. Find a way to destroy the pathogen that has this antigen on it. So it's relatively similar in that way to helper T cells. There's a lot of overlap in all the immune functions, and we're not going into all of it, and that is A-okay. But that is generally how those things work. Sound good? Okay, what questions do y'all have on activity 1? Nada? Okay, let's go ahead and split back into the breakout rooms. We'll cover add-on 10 and then we'll cover the rest of them. Hopefully we'll be able to get through it by the end of the session. There is just so much and so much detail that I think takes further explanation. So hopefully we'll get through all of it relatively soon. Let's say meet back here at 7:40, 7:45, and then we'll go from there. Sound good? Okay. I will see y'all back in a few. Start the recording. All right, sweet. So let's jump into activity 2 now, talking about some memory and antibodies and things. Starting off with number 10, how does one B cell or T cell turn into multiple cells?
I had this one. So I think, and I mean I think, they go through multiple cell divisions, so they become like a clonal section. So basically it's like a big like group of identical cells, and then they can either turn into effector cells, which help act against pathogens, or they can become memory cells, so that they won't forget the antigen if it's encountered again.
Awesome, yeah, exactly. So you start off with 1 or 2 of them, you go through clonal selection, which is like, just think of the name, you clone it, and then you select for like the best ones. That's kind of what we talked about earlier with the house keys. You make a bunch of keys and then you figure out the best one, and then you select, so you're selecting from those clones, and then some of them are effector cells, which are cells that actually go and fight, and other ones become memory cells that you just hold on and save for later. Okay, nice, Sydney. Number 11, how does immunological memory work? Who had 11?
I had 11. So basically what it does is it makes sure for the long-term run that you can be able to fight off infections. So there are two like sort of steps of how this works. Is the primary immune response, which is when your body first encounters a pathogen and it secretes those antibodies. And then of course, sooner or later that pathogen will get destroyed. But those antibodies still are within our system. So that way, if we were to somehow become infected with that same pathogen later in life, that's what's called the secondary immune response, which would be in a much quicker response because we've already secreted those antibodies. And so that way the pathogen would die quicker.
Dude, spot on. Yeah, exactly. That primary immune response, it takes a while because we have to figure out what antibodies to make and to find the best ones. That takes a while. And then when we have that secondary immune response, we've already got the good antibodies. We just have to make a bunch of them. And then you have a stronger and a faster response. Nice, Raj. All right, hopping on to number 12. What is the difference between humoral and cell-mediated immune responses? Who had 12?
I did.
Okay, sweet.
Okay, so the humoral response involves the activation of B cells, and that leads to the production of antibodies, which defend against bacteria and viruses in body fluids, like the blood and the lymph. And then cell-mediated is when specialized T cells destroy infected host cells, and that's all I really had for that one. But they both include first, primary, and secondary responses.
Yeah, nice, Katie. Yeah, and the humoral, this is kind of it's an unfamiliar name maybe, but if y'all remember the old Greek theory about the human body, that there were 4 humors, aka 4 things in the body that led to you being healthy or not healthy. Some of them were like blood and other things like that. That's where this humoral is coming from. So it's just like, think of like antibodies in fluid. So antibodies inside blood and inside lymph. That's kind of where that's coming from. And then yes, as Kate mentioned, the cell-mediated part of this is T cells destroying cells, some of our other cells. This is the cytotoxic T cell thing that we talked about a little bit earlier. Some T cells are helper T cells that help activate things, and other ones are cytotoxic T cells that specifically attack and destroy our own cells after they've been infected. But they need the help of the helper T cells to make sure that they don't just attack and destroy every cell in our body. Okay, solid. 13, how do helper T cells work? Oh. Who had 13?
That was me.
Nice.
So we also said, adding to that, basically when a T cell, or I think even a B cell, like responds to a pathogen, then they go back to the lymph, I don't know.
Lymph nodes? Yeah.
Yeah, and they like signal for other like cells to help because maybe they can't do it alone. So we like described it as like having an issue by yourself and then you call your friends. It's like kind of like the same way.
Yeah, that's spot on. And then when they go back too, not only are they getting help from their friends, but they get even better friends because they get more and more specific antibodies every time. Nice analogy, y'all. Okay. Any questions on 10, 11, 12, or 13?
So the helper T cell, so it's a T cell that goes to the lymph nodes, right? Not like the macrophage.
Yes, the macrophages are floating around in your body. The helper T cells go and find the antigen on your macrophage and then take that antigen back to your lymph node, which you can think of the lymph node as kind of like the factories in a sense. They're the ones where all of the clonal selection is happening. They're the places where all this positive and negative selection is happening. They're the spot where you're making better antibodies and occasionally worse antibodies, which just get trashed, thrown out. But that's where a lot of this complicated stuff is happening. And within the lymph node, there are all sorts of small parts and sections, and everyone has their own little like office space, if you will. But generally speaking, that's where most of the interesting parts of immunology are happening.
Cool.
Yeah. Other questions? No? Oh, tears. Okay, well, we'll move on then. make sure to please ask questions if you have any, because I know there's lots of confusing stuff going on here. So, okay, 14. How do those antibodies that we keep talking about actually do their thing? How do they actually help us fight the pathogens? Who had 14?
I did. So the way the antibodies actually work is that the antibodies themselves don't destroy the bacteria or virus or whatever the antigen is. So the way it works is that when the antibody binds to the antigen, it neutralizes the virus or the whatever it is, neutralizes the antigen. So when it does that, it can't replicate and it can't infect other cells. So it's like neutralized and it can't do anything. And then after that is opsonization, which is when it presents the antibodies present the antigen, the virus, whatever, to a macrophage, and the macrophage eats it. Phagocytosis happens.
Yeah, awesome. Yeah, mainly there's two ways, that neutralization and opsonization. I like to think of neutralization as like getting handcuffed. You're not actually destroying the pathogen, but you're somehow making it unable to do stuff. So that could mean, and usually that means that the antibody's binding to a protein on the outside of the pathogen, and that protein used to be really nice for helping the pathogen slide into your cells or to multiply or something, but that capacity has been neutralized. It can no longer do those things because there's an antibody bound to it. Whereas the other part of it, as Will mentioned, opsonization, is more like when it binds and it sets off a flare and it says, hey, come phagocytose me, come destroy this cell, which actually leads to cell destruction, whereas neutralization just leads to like a stoppage of the protein usage. Yeah, well said, Will. Number 15, what are the different kinds of antibodies? Who had 15?
I did. So with the different kind of antibodies, there's 5 different kinds, and they're like GAMED, or known as like IgA, Ig, so-and-so. And IgG is the only one that is membrane bound, while the others are just soluble antibodies that are like tear, saliva, and other stuff.
Nice. Yeah, and I just threw something in the chat that y'all can probably see. You'll see a lot of things written like this, IgG or IgA. That the first Ig stands for immunoglobulin, the last one stands for the letter of what it is. So as Haley was saying, there's 5 different ones. Yeah, there's not a lot that y'all need to know other than that membrane, or IgD is membrane bound, i.e. it stays on the membrane of something, whereas all the other ones are soluble, which is a fancy term of saying they can get secreted, they can dissolve, not really dissolving, but they're floating around in the fluid. So they call it dissolvable or soluble. It's not that it breaks up in the fluid, it's just that it floats around and is active in the fluid. And then there's a nice acronym for all 5 of them, GAMED, G-A-M-E-D. I think Haley was saying, that you can use to remember them all. And there are differences, but again, y'all don't have to worry about them. So no need to sweat it. Cool. Do y'all have any questions on 15? No, relatively self-explanatory. Okay. All right. Number 16. We talked about this a little bit already. 16, how does the complement system actually destroy pathogens? Who had 16?
So I had 16. I'm kind of adding on to what we said last time. So it uses a membrane attached complex that forms a pore in the membrane of the cell, like we said before. So ions then go into the cell, causing it to swell, and then it lyses, and it stimulates phagocytosis, which destroys some pathogens.
Nice. Yes. One thing that happens as it's pouring or having all the pores happen, and it can get phagocytosis too. But yes, the main part is just as you mentioned, forming that membrane attack complex, which forms the pore, causes water and ions to flood inside, and then it swells and lyses. So nice. Thank you, Sarah. All right, number 17. Okay. Now we get some fun ones. What is the difference between active and passive immunity? Jenna, you had this one right.
So active immunity develops like naturally, but I mean they're both natural, but via like lymphocytes or immunizations. So the pathogen is getting directly into the body, or they're getting like part of it, like in vaccines, and then.
I did the passive. So passive is when the antibodies are produced by someone else. So this is when like there's a mother that's pregnant and then she passes the antibodies through the placenta to her fetus or through breast milk. And then this only lasts for like the amount of time that the antibodies are actually being transferred to the child. So it's only for like a few weeks or months, whatever. And yeah.
Nice. Yeah, I really like how you said that. You're not making the antibodies yourself in passive immunity. Someone else is making them and giving them to you. So like with active immunity, you're getting the pathogen, whether it's injected or infecting you, or the protein from the pathogen, and you have to actively figure out what to do with this. How do I fight this? What's going on? It's a very active process. But in passive immunity, someone is just giving you, whether it's through the placenta or through breast milk, or you can get like an injection for like a snake, like an anti-venom thing. Someone's just giving you the antibodies and then they just do their thing. You're not really doing anything to get them. And the reason why why passive immunity doesn't last as long is really just that the antibodies break down over time throughout your body. And so once they all break down, you're not making them, so you don't have them anymore. Whereas in active immunity, you made them in the first place, so you can keep making them if you need them. But you can't do that for passive immunity. What questions do y'all have on 17? I'll also add the vaccines can also be both. So a lot of vaccines, excuse me, they inject you with they'll call it the attenuated, which is it's the pathogen that's not dead, but it just can't infect you. So it's attenuated. Or they'll inject you with a dead pathogen. That would be active. There's also passive, like vaccines that function by passive immunity, where you're injected with like antibodies from a cow to smallpox. I think smallpox is one of them. So they inject the cow gets smallpox, develops antibodies against it, and then you take those antibodies, inject it in a human, and that functions as a vaccine. And in that particular instance, the human can use those or replicate them somehow. But those are more hairs to split when you take actual immunology. But vaccines can be both, although they're typically active.
Is that what they're doing for the coronavirus?
That's a great question. I have no idea what they're doing. I feel like every time I look up something or I hear an idea, it's just one thing is opposite from the other. Not even sure the test. I'm really curious to look at the test though, because the coronavirus itself is just from that crown-shaped corona family of viruses. You know, I've heard about all the people who are asymptomatic to it, like they'll get the disease, but they don't get any symptoms. One possible cause of that is that they've been infected with another coronavirus. And so they already have an immune response to that coronavirus. And the new, like COVID-19, looks similar enough that you can fight that one with your old stuff. So it's possible that these people have already gotten another coronavirus, but a very common, just whatever infection, nothing serious. And then when they get COVID, they've already had something similar to it, so they can fight it better. That could just be me making. I mean, that's how some immunity to things work. It could be that they have other things preventing it too. But as far as I know, the scientific world does not yet know why some are asymptomatic or why some are not, or exactly how they would do that. But these are just some possible ideas. Hopefully soon we'll know, but till then. Good question though. What other questions do y'all have on 17? Okay. let's move on to 18 then. What happens when an immune response is too strong?
I had the answer to that one. So when an immune response is too strong, it is usually because it's reacting to an allergen, and so the response would be a person's allergies. Um, this can cause a person to go into, I think, anabolic shock if the response is too strong. Another thing that can happen is an autoimmune disease where the immune system will attack the body's own cells. And so an example would be like diabetes, where the immune system will attack the beta cells, and now the body cannot produce insulin.
Nice. Yeah. So the allergies, so when your body has a really, really strong immune system, how do I want to say this? The immune system has a lot of places where you want balance to it. If it's really, really strong, you're going to attack your own cells or overreact to stuff, which can lead to diseases. And if your immune system doesn't work, you're going to have other issues, which we'll talk about in a second. Allergic reactions are just your body interpreting the little proteins in like tree pollen as if it were a pathogen. So all of those allergens are really just antigens. They're just little proteins or whatever, but your body for some reason says, ooh, that looks too similar to one from a disease or from a virus or from a bacteria that caused a disease a while back. So we must be scared of this and we must react to it. And that's why you get like a runny nose. That's why your airways like shut and all that, because you want to make sure not to get that pathogen into your body, which can cause anaphylactic shock, which is why you have to have the EpiPens and all that kind of stuff. And one example of an autoimmune disease, like Cole brought up, is having like type 1 diabetes, where something attacks your beta cells, then you no longer have cells that produce insulin, and then you have to have a pump or something like that. Now the opposite, which this is confusing because on the same slide that talked about what happens when an immune response is too strong, there's also a bullet point at the bottom that talks about immunodeficiency. Please do not get confused. Immunodeficiency is the opposite of having too strong of a response. Immunodeficiency is what it sounds like. You have a deficient immune system. So immunodeficiency is when your immune response is too weak. So this is when you're extra vulnerable to infection. This is like your friend that has to that gets like strep throat like 7 or 8 times a year, or who's always getting a cold and has to spend a week out from school because their body just can't fight it off. Whereas most of us might get sick or have a sore throat for a day or two, but then we're fine. These are people who just can't fight it off well. And if you're really immunodeficient, well, you can have all sorts of troubles and diseases. You could also be more vulnerable to cancers. But cancer is really complicated. There's a lot of stuff going on with that. If you're interested, you can talk to Dr. Tauby. He teaches the immunology class, and he does research on breast cancer. He actually works with one of the OChem labs, and the OChem lab develops potential chemicals like basically chemotherapies. And maybe that's a poor use of the word, but they develop chemicals that can help fight those breast cancer cells, and so they work together. So a really cool lab. I've had a couple friends work in it, so y'all should hit up Dr. Tauby. He's also just a really sweet guy if you've never met him. So, but anyway, that is all for the worksheet. Sorry, we are a couple minutes over. Feel free to head out if you need to. This is, I guess, our last regular SI session, which that's kind of sad. But before I start crying, y'all are welcome to head out if you need to. Please stay around if you do have more questions, and then Katie and I will be updating you. I think the plan is to have sources like the PowerPoint and practice test hopefully available like Sunday, maybe Monday, and then Monday night I'll have some office hours to talk through, probably like just 2 hours straight. We'll just sit down and talk through all of the questions that y'all may or may not have, and that way you'll then have Tuesday and parts of Wednesday at least to study, and then for the other section y'all will have more time. So okay, yeah, I think that's all I have. If you're heading out, have a good night. Hopefully I'll see you in office hours. If not, best of luck on the final. I'll be praying for y'all. Hopefully all goes well. And then if not, please do show up to office hours and we can keep talking science and stuff. So
Thanks, Liam.
Hi,
Yeah.
thanks,
Thanks,
Lena.
Liam.
Have a good night.
William.
Bye.
Bye, y'all.